When talking about the diversity of life, a lot of people like to talk about life as being either eukaryotic or prokaryotic. Eukaryotic life has a membrane-bound nucleus as its fundamental defining characteristic, and so the eukaryotes are the plants, animals, the fungi, and the protists, where the protists comprise a bunch of single-celled non-plant algae, as well as heterotrophs and a whole diverse array of life. But they do all have a nucleus. And additionally, the eukaryotes are said to be all of the life that's multicellular and all of the life that has organelles on the interior of the cells, like a chloroplast or a mitochondria. On the other hand, prokaryotes are bacteria, which are single-celled, simple, and archaea, which are basically bacteria, although you've probably heard that they only live in extreme environments compared to where we live, or that they are older than bacteria, or some weird stuff like that. But in general, the prokaryotes are said to have simple cell structure and organization. So they don't have a membrane-bound nucleus. They don't have any kind of organelles. They're basically, I guess, just water balloons. And I mean, I guess on the surface it seems like it's probably not too bad of a classification system, but when you start digging into the molecular structures present in different kinds of life, and the evolutionary history of life in particular, it turns out that archaea, while they are single-celled, they aren't more similar to bacteria than they are to eukaryotes. Archaea are actually a lot more similar to eukaryotes in a lot of really fundamental ways than they are to bacteria. Now, when you start comparing life and trying to group it into sets of life that are more similar to themselves than they are to the rest of life on Earth, it's important to look for structures that are found in all of life, but that are diverse throughout life, so that you actually have some resolution in finding differences between different lineages. Now, one such structure would be a membrane. All life, at a minimum, has a plasma membrane that separates its cytosol from the environment. This right here is a diagram of a section of a phospholipid membrane. You can see that it has an exterior surface that is polar and interacts favorably with water, which is the solvent that life takes place in. And then it has an interior surface composed of hydrophobic lipid tails. And then within this membrane, you have different selective channels, so that the membrane is selectively permeable, but that not just anything can get across. This allows the cell to keep a stable interior, maintain homeostasis, in order to make sure that its metabolism is energetically favorable, and all that kind of stuff. Now, in the case of this eukaryote-prokaryote division, you would expect that the membranes of prokaryotes are more similar to each other than they are to membranes of eukaryotes. But what I have here is a series of figures describing the structure of the phospholipids present in the different membranes, and what you see is that eukaryotic and bacterial membranes are very similar. They have a glycerol molecule, which is a 3-carbon molecule, each carbon having an alcohol group. To 2 of these alcohol groups, you have fatty acids. So these are 2 examples of fatty acids, all of which share this carboxylic acid group here, and then a chain of carbons that can be it's variable in length, and it's also variable in its saturation and its orientation. So you can see here trans oleic acid and cis oleic acid. It's the same number of carbons, the saturation is the same, but then the structure of this double bond here is different. In any case, you have 2 of these, each one bound to this glycerol by an ester, and this group right here is an ester group. The R represents this variable chain. And then on the 3rd carbon, you have a phosphate group bound to a carbon. And this doesn't change. And so you can see on this side, we have archaeal membranes. And this 3-carbon group here is the same. This isn't showing the actual phosphatidyl group. It's not showing the phosphate group on the outside. But this hydroxide here would just be this phosphate. And it looks similar, but if you pay attention, this chain here is actually branched. These are isoprene side units. This right here is an isoprene subunit, and these double bonds can be polymerized to form these chains. And so instead of this unbranched fatty acid, you actually have a branched isoprenoid chain here. And in addition, archaea don't have ester bonds between these lipids and the glycerol. They actually have ether bonds, which you can see here. This is an ether bond, ROR, which is differentiated by this carbonyl on the ester. The ether doesn't have that carbonyl, and so it's actually this R right here is going to be the carbon with the carbonyl. And so the ether bond, for reasons I won't get into, is more stable. Additionally, some archaea don't actually have a phospholipid bilayer. They have a phospholipid monolayer, which is a 4. Oh. And so instead of having 2 lipids with 2 ester bonds or 2 ether bonds to a glycerol, they have 2 lipids with 4 ether bonds to 2 glycerols. And so additionally, they can have a cyclic phospholipid where this one chain is bound at the end. And in both of these cases, I guess D and T, the membrane has additional stability in high temperatures. That, together with this ether bond, allows archaea to be a lot more stable in particular environments. And more importantly, it means that when you look at the membranes, archaea are not more similar to eukaryotes than they are to bacteria. And bacteria are more similar to eukaryotes than they are to archaea. This goes against this eukaryote-prokaryote dichotomy.